So in this video, I'm going to be talking about the basic structure of an amino acid. Cells produce tens of thousands of distinct proteins. They're vital and versatile components of the cell. And those proteins are made up of monomers called amino acids. And proteins and amino acids were most likely abundant during chemical evolution. Most proteins are made up from a combination of just 20 amino acids. Each amino acid, each monomer, is composed of a central carbon bonded to a hydrogen atom, an amino functional group or carboxyl functional group, and an R group, which is a variable side chain. And we'll have a separate podcast that's going to address these R groups. So here's the basic structure. Here's our amino group. Remember, that's a nitrogen bound to hydrogen. And here's our carboxyl group, right? A carbon with a double bond to oxygen and a bond to a hydroxyl group. And these two, this amino group and this carboxyl group, are joined to a central carbon. And that central carbon has a hydrogen and it has a side chain that we often represent as R. So this is the basic structure of an amino acid. But as we talked about when we talked about functional groups, this amino group tends to act as a base and gain a proton, and this carboxyl group tends to act as an acid and donate its proton. So the ionized form is that this amino group picks up this hydrogen here, so the nitrogen has a positive charge, and this carboxyl group donates the hydrogen, so this oxygen has a negative charge. And more often than not, this is the version that's found inside our cells, which is a good thing because these ends are highly reactive, so they'll participate in forming peptide bonds. So proteins are macromolecules, which means that they're large molecules made up of smaller subunits. And the smaller subunits, which I talked about, are called monomers, or one part. And monomers link together in a growing polymer, okay? Polymer meaning many parts. And lipids happen to be the only macromolecule that do not form polymers. So we'll talk about nucleic acids and carbohydrates. Those are polymers, and lipids are the only ones that are not. But let's return back to proteins. So amino acids are the monomers that make up proteins, and proteins being the polymer, right? So here's the growing chain of our polymer. Monomers polymerize, they become polymers through condensation reactions or dehydration reactions. The result of this is loss of a water molecule. So a water molecule is going to come out of the molecule so that a monomer can join. And hydrolysis is the reverse reaction. So this breaks apart a polymer. So these reactions tend to be less energetically favorable than a hydrolysis reaction that would break apart a molecule. Polymerization requires energy, so monomers would not self-assemble into a polymer, and that's because of entropy. So polymerization decreases entropy, which essentially is the disorder of a situation, and so because of this, it's not spontaneous. So you often need energy. So during chemical evolution, polymerization would only have happened if the amino concentration was very high, which it was. And several mechanisms may have led to polymerization during chemical evolution. So we might have gained that energy to create macromolecules. So here's what that reaction looks like, just very basically. We have monomers, and what's going to happen in a condensation reaction is the monomer is going to go in, it's going to join the polymer, and water is going to come out. In a hydrolysis reaction, we're going to put water into the molecule, and the monomer is going to come out of the polymer. So water in, monomer out. And here's what this looks like in a peptide. So the bond that forms during a condensation reaction between two amino acids is called a peptide bond. And this peptide bond is formed by a condensation reaction. So here is our ionized amino acids. We have two amino acids here. We have our amino group and our carboxylic acid in both of these. And what's going to happen is this oxygen on this carboxylic acid is going to come out with these two hydrogen, right, in a condensation reaction. So those bonds are broken, and water goes out. And when water goes out, these two monomers are joined. And that bond, that covalent bond that forms there, is called a peptide bond. So there is our peptide bond, right? So it's between this double bonded oxygen and the nitrogen of the next amino acid. So a chain of fewer than 50 amino acids is called an oligopeptide, or a few peptides, or just a peptide. And a chain of more than 50 amino acids is called a polypeptide. Proteins are the complete functional folded form of a molecule, and sometimes used to describe any chain of amino acids or residues. But for the most part, in this podcast, we're going to be talking about oligopeptides. So here is an example of an oligopeptide. So we've joined a couple amino acids together. And looking at the structure, I want you to pause this video and see if you can identify how many amino acids are in this oligopeptide, how many peptide bonds are there, where are the R groups, at what end does the peptide start, and what end does the peptide finish. Okay, so how many amino acids are in this oligopeptide? So first, let's count the amino acids, right? So we have, here is our amino group, and there's what's left of our carboxyl group. So here's, essentially we can count at every oxygen that's pointing up. 1, 2, 3, 4, 5 amino acids in this oligopeptide. How many peptide bonds are there? So the peptide bond is formed between the oxygen, right, that double bonded oxygen, and a nitrogen, right? So when you see these two together, you can count a peptide bond. So 1, 2, 3, 4 peptide bonds. So there should always be one less peptide bond than there are amino acids in a peptide. Where are the R groups? So the R groups are bound to the central carbon. So here we have our amino group and our carboxyl group, and here's the central carbon. So this down here would be our R group, right? This hydrogen here. So this is actually glycine here. So again, let's find our nitrogen, central carbon, our carboxylic group. So this is the hydrogen that's part of our R group. Here's another R group and another R group, right? The carbon that comes right after nitrogen, and another R group. What end does the peptide start? So all peptides, because of the way they're joined, have directionality. So all peptides start at the amino group, or what's called the N terminus, and they end at the carboxyl group, or the C terminus. And so proteins are read in the direction from, when we're saying what the sequence is, from the amino group, right, to the carboxyl group. And because of this way in which peptide bonds are created, we end up with a few things. We have this R group orientation where all the R groups are facing in one direction. And so, as you'll see in later videos, these can interact with each other. We have directionality, which I talked about, right? So we have this N terminus here that starts the protein, and the C terminus that ends it. And we have flexibility. So there's some rotation that can occur around these bonds here. So you have a little bit of flexibility in that molecule. So here's just, as I told you before, all the amino acids you were looking at were glycine. And so here is a chain where there are other R groups. Oops. So here's glycine again. And we have other R groups that are present as side chains on this amino acid, right? And they're joined between the carbon and the nitrogen by a peptide bond. And so all of these side chains can be represented as a 3-letter code as well. So we have our N terminus beginning the protein, and then glycine, proline, serine, and so on until we get to our C terminus. And so this one at the N terminus was 1, and the last peptide, or the last amino acid, is 8 in this oligopeptide. So in summary, amino acids have an amino group, a central carbon, a carboxyl group, and an R chain off that central carbon. Peptide bonds join together amino acid in a condensation reaction, so water is lost. And peptide bonds have directionality. Because of that, we have orientation of our R groups, and this gives proteins a flexible backbone. So that's all I have to say about the basic structure. In the next video, we're going to talk about those side chains in more detail.